All right, hi everybody. Thanks for your interest in this session. It's really a privilege for me to speak with you all today. So I have the job of expanding from our infant applications into brain development in general and links to behavior. So let's get into that. Obviously, there's a lot of work to summarize in this field, so I'm going to be kind of talking in broad strokes for the most part. My goal for this talk, so you know, coming into it, is kind of to give you an idea of what we know about brain development and links to cognition and behavior, and also identify some areas of opportunity if you're thinking about how you can build in this field. You know, what are some tools you could use to kind of have a big impact with your research. So we're starting from the point of having our nicely segmented major fiber bundles or highways in the brain, and starting to look into them with tractometry. So now that we have our tracts, we're going to do tractometry. And I just want to kind of start by calling back to Fan's video, where he had a great description of many different ways we can do tractometry, and highlight that, you know, the most basic and also the most common method that's used in the field is mean metrics across a whole major fiber tract. So if we have a region of interest, we've identified where in the brain this tract appears to pass through, and say, for example, I've got on screen a map of FA in the brain, fractional anisotropy, probably the most commonly used metric to quantify tract structure. What we're talking about when we're talking about FA of the splenium in my little visual example there is essentially just averaging the FA values of every voxel that tract appears to pass through. Of course, like Fan described, there's more sophisticated ways to do this, but mean and averaging across our voxels is the most common way to do this, and indeed it can tell us quite a bit about brain development. It's taken us quite far, I would say. So let's get into our general trends that we've discovered so far. The majority of work, as we've kind of seen in our previous talks, is based on the diffusion tensor model, so diffusion tensor imaging, using these metrics like FA and MD, which is mean diffusivity, and we have really well-established trends that have been identified, those being that FA is going up across development and MD is going down, kind of in a matched manner. We know that this development is non-linear, so we see quite rapid changes in these metrics in the first couple of years of life, followed by more gradual changes that can even progress into early adulthood, which we'll get into on the next slide. And another thing I want to note is that sex differences in development are typically minor. What I mean by that is that they're inconsistently reported. So sometimes studies will report differences between males and females, sometimes they won't, and sometimes they'll be in the opposite direction as other studies. So there's a little bit more cloudiness in that area, but I would say they're generally pretty minor. If I had to point out one trend that maybe is reported more frequently, you might see, earlier development or kind of further progression along these trends on screen for females than males, but that's mostly around puberty, I would say. Another really interesting trend that's well defined is regional variation in these trends. So another key feature of development is time to reach the developmental plateau, which is essentially when we just see these curves of change in FA and MD level out and become quite stable. You can see there's clusters of tracts that have been defined as far away as 2010 or 2011, something like that. We can see groups of tracts that are reaching that developmental plateau by early adolescence. That's that group on the left there. Still more tracts that are developing throughout adolescence and reaching those plateaus around 20 years old, and even tracts that are developing past 25 years of age. So things like the cingulum and the unsent fasciculus on screen are still undergoing gradual change as we're past our undergraduate degrees and in our full independence, which I find quite interesting. This also, I think it's a really interesting finding. It's one of my favorites, I suppose, if I had to pick favorites, just because it kind of gives us a set of guidelines to understand links to cognition and behavior as well. The general trend there is that tracts that are maturing are related to cognitive and behavioral skills based on, broadly speaking, the cortical regions that they connect. So if I can point your attention to the bottom figure on the screen, this is a study of change in some reading-related tracts, the arcuate fasciculus, which connects some major language regions in the brain, and the inferior longitudinal fasciculus, which is, going from our visual cortex into our temporal lobe and includes connections to some word recognition areas. So the general trend here is as those tracts mature, we see improvements in the cognitive or behavioral skills that they kind of underlie. Another example I've got on here at the top is a behavioral example where kids with lower externalizing behavioral scores are showing this increase in FA over time, while kids with higher externalizing behavioral scores don't show that trend, implying that there might be some differential development that could be underlying this behavioral presentation, or it might not. A couple other notes about the externalizing behavioral study. These are all healthy kids, so there's no behavioral diagnoses at play here, but still can be quite informative. And if you're not aware, externalizing behaviors, just for some context, are negative behaviors that are projected or directed outwards in the world, like hyperactivity or aggression. So the general trend here being, as our tracts mature, we see improvements in the cognitive or behavioral domains or skills that they underlie. Now, this kind of research has been going on for quite a while, which means we understand these trends very well, which is great. So here is a meta-analysis showing changes in FA or trends in FA in different regions per panel, and each colored line is a different study that's reported these trends. You can see in the little descriptors at the bottom, studies going as far back as 2007. We're nearly 20 years ahead of that, and we're seeing across our studies quite a bit of convergence. You can see lines that have the same slope reported across many of the studies, and also some interesting areas of divergence, places where our studies aren't really agreeing, particularly in terms of magnitude of FA values in this example, which I'm happy to go into in the question period if you're wondering why that might be the case. But my point here is that we have a pretty robust and consistent understanding of trends based on DTI metrics, FA and MD, at the whole tract level, right? Talking about, FA of the genu of the corpus callosum or FA of the corticospinal tract. This is a major highway that we understand development of quite well. So at this point in the talk, you might be thinking, okay, we've been doing this kind of research for almost 20 years. If I'm going to do a study, where does that fit in? Am I just going to be reporting the same thing everyone else has? So for the remainder of the talk, I'm going to talk about some opportunities to build on this really solid foundation of literature that we have to take advantage of. So I've got two opportunities, the first of which is the tract or region of interest, which several previous talks have touched on as well. So I want to start here with an example from some of my own work, where myself and a colleague, found just a correlation between FA in the splenium and math abilities. So we saw that individuals in adolescence that had higher FA of the splenium also did better on our tests of math that we made them do, that they were all very happy about. Just take a second to think about what that means. What does a correlation between FA in the splenium and math scores actually mean? This is a huge tract, right? It's a bilateral, connecting many different cortical regions, involved in many different types of processing. So it's probably not the case that FA values at each point of that splenium are related to math ability. It's probably just a more limited relationship that's being broadened across the whole tract. And indeed, when we followed up with an along tract analysis, which we've heard about before in our session today, we saw areas where this partial correlation was replicable, or it was significant in the along tract analysis, and areas where it wasn't. That's those shaded bars. Hopefully you can make them out from your seat. Those are areas where the partial correlation was replicated. If I just turn to my cursor here, the way that played out is our correlation was significant in this region where the splenium, fibers in the splenium were exiting the midline and entering into the left hemisphere. And this is an area where other major fiber tracts are passing through, which you can see I've drawn on my map in painstaking detail. So there's the inferior frontal occipital fasciculus in green and the inferior longitudinal fasciculus in purple. We've heard a little bit about how the diffusion tensor can't really distinguish between multiple fiber populations, which are present in many white matter voxels in the brain. And so some follow-up questions that we can't answer with this analysis on screen alone are, is it maybe the case that just some interaction between these tracts is really driving the relationship with math, and that's being played out in our FA findings? Or what if our relationship between splenium FA and math scores is actually driven by the portion of the IFOF or the ILF that we've captured in this area? With the tensor, we can't necessarily distinguish between those things, but they are interesting follow-up questions. And I've included a little quote from a 2021 paper noting that some tracts have more consistent relationships with behavior than others. They highlight the corpus callosum and the cingulum, which I think is quite informative because those are tracts that have predominantly one major fiber orientation, the cingulum going anterior, posterior, and not interacting with many other crossing tracts, and at least the core of the corpus callosum crossing the midline. All of our axons should be oriented medial-lateral as well. So that all, again, kind of implies that this interaction in multiple fiber populations can kind of mask behavioral relationships or certainly complicate the analysis. But we can go beyond the diffusion tensor, right? It does require us collecting more data, which, as you've heard, collecting more data is always better in this field anyway, so why not take advantage of that? And the most kind of common model that we can use to clarify things like multiple fiber populations, which we've already heard about, is the fiber orientation distribution function, which can be computed from something like constrained spherical deconvolution, which our previous talk already touched on. The advantage of this model is that instead of all of our fiber orientations and the diffusion along those fibers being lumped together into one ellipsoid, like you can see kind of at the bottom of the screen here, instead we can have a model that represents diffusion in multiple different orientations. So that's our FODs at the top here. You can see multiple lobes of diffusion in different orientations, and we can presume those are most likely representing different fiber populations in a voxel. So with a model like the FOD or other interchangeable acronyms, we could begin to distinguish between areas where, say, we have an intersection of the corticospinal tract and this arc fasciculus or SLF that I've got in a little cutout here, which would be very useful to help us to disentangle relationships like I had on the previous screen where we've got multiple fiber populations in the same voxel. So that's one thing to consider. And another advantage of using it as a kind of model that represents more detail than the diffusion tensor, which kind of lumps everything together, is that it unlocks new kinds of analysis, like pixel-based analysis. If you haven't heard of pixels before, these are representations of different fiber populations. So you can have multiple pixels in a voxel to represent those different orientations of diffusion. And with an analysis type like this, we can begin to look at tracts separately. So I can show you in this bottom right little panel here, we have area or voxels in the corpus callosum that have one pixel, but once we get to this region of white matter where there's an intersection with the cortical spinal tract, you can see two pixels, which means we can look at those fiber populations separately and begin to clarify our DTI-based understanding of development and links to behavior, which is great. However, because we're not using the diffusion tensor anymore, we don't necessarily have access to FA and MD. We could, but that wouldn't necessarily provide us the advantage of looking at pixels separately. So we turn to new metrics instead, which can help to clarify FA and MD-based relationships that we understand quite well. So pixel-based analysis offers metrics like fiber density, fiber cross-section, and the product of the two, or FDC, which would be closest to FA, I would say, because it's kind of lumping multiple features together. And I'm going to take this opportunity to direct your attention to the genu and the splenium, the genu being the front of the corpus callosum here, and the splenium being the back of the corpus callosum, which I've already talked about. In our FDC metric, we see changes in both of these regions, and this is a longitudinal study in 9 to 13-year-olds, which is, kind of analogous to our increases in FA. We know there's development occurring in both of these regions, but with our kind of more specific metrics, we can see changes in fiber cross-section in the splenium, but not in the genu, and changes in fiber density in both. And this begins to give us a more nuanced understanding of what's driving our FA-based relationships. We can say, okay, in the splenium there's changes in fiber cross-section and also in something like axon packing or diameter, which might be driving these fiber density results. Whereas in the GNU, we see development, but it's largely through fiber density changes and not so much fiber cross-section. So perhaps that means these tracts are at different developmental stages or that they're developing differently. But either way, we're beginning to kind of uncover new detail in these relationships and in our understanding of white matter development. I'd be remiss if I didn't talk about connectome-based or graph theory-based analyses as well, which Fan thankfully laid out for me nicely. So I've just got a couple key findings here to talk about, and I think this is an important method to consider because we know that our brain operates through these distributed functional networks that are coordinating processing through our white matter tracts to actually execute information processing and produce behavior in the first place. So something like understanding or investigating a connectome might better represent what's actually driving cognitive and behavioral processing, which is very informative and might kind of, it certainly provides us a different paradigm than our whole tract-based analysis, which I would say is most common. So in connectome-based analyses, we can look at network architecture and the properties of the network underlying our functional networks. We can consider, the architecture of that network. Our brains are typically arranged in kind of like a small world network architecture where we have nicely interconnected networks that are connected between each other by these hubs or rich club regions. Through connectome-based analysis, we know that architecture is present at birth and it refines into adulthood through things like increasing efficiency. So the paths between regions tend to get shorter, and connections within networks tend to become more clustered, while connections between networks tend to become less clustered or kind of trimmed over time, and those reliance on those hubs connecting our networks increases as those hubs become more interconnected as well. So another really useful way to look at development that goes beyond our kind of whole tract DTI-based understanding of brain development. And also, this connectome-based approach is, connected in previous studies to our FA and MD-based trends. So there's certainly some convergence here, and potentially an opportunity to view the brain in a new light and kind of go in a different direction in this field specifically. So there's some food for thought in terms of how we consider our regions of interest. And now let's talk about the metrics that we use. Like I mentioned before, FA and MD are the most common metrics analyzed in this body of literature. And whenever we're talking about them, we have to kind of be careful of the language we use because there's all kinds of different factors at play in each of these metrics, both biologically meaningful and experimentally determined, which that's a whole other can of worms that I don't have to talk about. But let's talk about the metrics. So the strength of DTI metrics is that they're sensitive to many different aspects of white matter microstructure. You can see my little circle with a bunch of, just a handful of different features that FA, MD, and the other DTI metrics can detect differences in. But the weakness of that, it's kind of a double-edged sword here, in that we're able to detect changes, but we can't determine which microstructural features are really driving those changes, which is quite important, especially if we're looking for biomarkers that we could potentially use to identify altered developmental trajectories in neurodevelopmental disorder or something like that. So this prevents us in DTI-based analyses from kind of answering more nuanced questions than just broad trends. So if we see an increase in FA or a decrease in MD and we want to know what's driving those trends, it's really hard with just DTI metrics to dig much further. And we also, this plot here is showing kind of smooth lines of longitudinal change across development. But if you narrow down to an individual subject, you'll frequently see quite a bit of variability between different scans or different time points for each subject, and also distinct differences between subjects that could be very interesting. But because our FA and MD metrics are so general, we can't necessarily dig into those too deep. But fortunately, there's been plenty of work to develop more microstructurally specific metrics that we can take advantage of. So if I just take myelin from that one circle Venn diagram, there's a nice meta-analysis that can visualize different metrics that are available, that are sensitive and variously specific to myelin. Might be kind of hard to make out, but each of these columns of colored dots is a different imaging metric that's sensitive to myelin, and the y-axis there is effect size, or R squared, reported by each of the studies. So you can see there's various levels of specificity to myelin, across these metrics, with each dot being a different study that use that metric, of course. And there have been analyses to kind of compare across these metrics and show that they tend to agree quite well. That's that scatter plot on the right there. The yellow voxels, this is a whole brain kind of voxel-based approach. We have a scatter plot here of two myelin-sensitive metrics with gray matter voxels in teal and white matter voxels in yellow. So you can see there's a lot of convergence between these metrics, especially in white matter, but there's also areas where the metrics are behaving differently. Take, for example, this cluster of gray matter voxels that there's a lot of variance in our myelin volume fraction metric, but our quantitative IGMT metric doesn't really, is more consistent in terms of the spread on the x-axis there. So that's just kind of to visualize that these metrics have varying strengths and weaknesses, varying additional sensitivities to microstructure in the brain, which might kind of inform when we want to use one metric but not the other, instead of trying to apply all 30 of these metrics at once or something like that. So we can use more microstructurally specific metrics to dig deeper and clarify our DTI-based understanding of brain development. And we can do that because, like I mentioned before, tractometry in its most basic sense is just averaging across voxels in the brain. So that we don't have to necessarily use a diffusion-based metric or a DTI-based metric to quantify tracts. All we need to do is have a tract mask and a whole brain map of some structural metric that we're interested in. And then we can look at that structural metric either in mean or in a long tract analysis or something like that, in whatever region we like. So this is a big strength of the technique. It's very flexible to any imaging modality or metric map that we're interested in. So I've got an example of how these metrics have been used to clarify trends in brain development. This is a study that I led looking at adolescent brain development over a 2-year period, for the most part, in many major white matter tracts in the brain. So in all of the tracts, which is it's 11 major white matter tracts investigated bilaterally, in all of those tracts we saw increases in FA and decreases in MD over our 2-year period. So great. We have confirmed again in adolescence that white matter is undergoing continued development. But because we applied some more microstructurally specific metrics, we were able to identify tracts that were developing in different ways. So on the left, we have a cluster or a group of right hemisphere white matter tracts where we saw increases in axon density. That's neurite density index from the NODI model, if you're familiar with that. We have a big collection of tracts in the middle there where we saw those changes in axon density and also changes in myelin content. And then in our uncinate fasciculus, which, if you remember the beginning of the talk, is one of the tracts that undergoes most prolonged development, we also saw changes in the G ratio, which is a ratio of axon diameter to axon diameter plus myelin thickness, or just fiber diameter in general, and kind of touches on communication speed between regions and changes in kind of efficiency. So including additional metrics beyond just DTI has allowed us to identify different trends in development in adolescence, and is, I think, really a promising direction moving forward. Now, there is, I have lived experience with this. The downside of investigating many tracts and having so many metrics available is that we can easily expand our statistical plans beyond what's reasonable. We have to be able to handle the family-wise error rate increase from running many different statistical models, and indeed that was a major problem in the study that I had on the previous slide. So one potential way that we could go beyond or account for this is maybe by looking for areas of convergence across our different metrics that share microstructural sensitivities, like myelin sensitivity or any other microstructural metric that we're interested in. Look for areas along a tract or maybe across multiple different whole tracts where these metrics are agreeing. There's many different ways to do this, but the example I'm going to walk us through today is dimensionality reduction through principal component analysis. There's a really nice study here that lays out the method in quite a bit of detail. It's a really great reference. What they did here was take 10 diffusion-based imaging metrics in many different white matter tracts in the brain. You can see them all laid out in this section of their figure here. Sorry, I want to get a circle going. And using principal component analysis and kind of along tract profiles, dumping that all into a PCA, they identified 2 components, which explained 80% of variance in their 10 input metrics. So they're retaining most of the meaningful variance in their 10 metrics that they had in their input sample, but collapsing that down just to two latent variables that they can use in their statistical models, which is going to drastically reduce the number of comparisons they need to run, or the number of models anyway. This expands beyond just diffusion-based metrics. So all 10 of these, like I mentioned, were based on diffusion tensor imaging or kurtosis or something like that. I ran a follow-up including some multimodal sequences, so not diffusion-based, like magnetization transfer and VCG ratio there and water fraction, and found, again, components that explained 80% of variance in our model, but instead of just the 2, there was 3. We interpreted 2 of them as having the same kind of sensitivities as the previous study, so tissue complexity or kind of fiber architecture, and a myelin and axon packing or a restriction-based component. We also identified another factor of axon diameter, which it could be an area of interest for us. Now, both of these studies showed that our principal components or our latent variables were sensitive to age effects, so we can use these to identify trends in brain development and again begin to clarify FA and MD based relationships and potentially behavior. I was looking at relationships with reading in adolescence, and we found some trends that I have on screen here, although they didn't survive multiple comparisons corrections, so that's worth noting, but it shows potential for further development to clarify our understanding of cognition or behavior as well. And of course, there's other ways to do this, perhaps through machine learning or deep learning based approaches, if indeed you have enough data. But I think being able to collapse shared information across metrics is a really promising way to take advantage of the range of different imaging metrics that we now have available to quantify white matter microstructure instead of just our broad FA and MD variables. So I'll wrap up there and summarize the talk by saying we have a really good understanding of white matter development trends across the whole developmental period and the general trends in links to cognition and behavior, which I hope I've established for you. The majority of this is based on the diffusion tensor, which is great. It's very useful. Obviously, it's been used a lot in the past. And we can go beyond this by implementing more complex representations of fiber populations or more microstructurally specific imaging metrics. And as the field kind of expands in the range of possibilities, it's worth considering that using multimodal analysis methods and including these specific metrics and also being able to kind of collapse shared information and look for areas of convergence are likely to be very promising directions moving forward. So with that, I'll thank the wonderful mentors and teams I've worked with over the years, our funding sources, and I'm happy to take any questions on the app. Thanks.
Okay, thank you. So we have a few questions here. Can you comment on how interpretations of diffusion metrics may be different across different ages?
That's a good question. I suppose it depends on, I guess the first place I would go is previous literature in that age range to look for commentary or maybe use of those more microstructurally specific metrics to see what we already know about development. For example, we have a pretty good understanding of myelin change. We see more rapid change during infancy than we do during adolescence, for example, and a lot of that is based on work in magnetization transfer or water fraction based or those metrics were used in studies that have identified that. So that might be able to kind of inform our understanding of what's driving change in FA and MD. That's the kind of technique that I'm most reliant on when I'm doing that kind of work. Yeah, I think that's where I would go as a starting place. Yeah.
On the one hand, you told us that FA and MD develop in tandem. On the other, they seem to weight on different principal components. Is that in conflict, or are both things true at the same time?
Great question. Really good, really ripe area for discussion. I guess I would start by saying that FA and MD do share a lot of information, but they also capture slightly different kind of aspects of diffusion, which means that they have they capture different microstructural properties. At least I would hypothesize, or I would argue. So I think both can be true at the same time, in that we're seeing, say if I just kind of give a little toy example for us to play with. Say FA represents myelin and MD represents axon packing or axon density or something like that. We can concurrently see changes in both. We can see increases in FA representing myelin and decreases in MD representing axon density. But if we have additional metrics which are sensitive to those properties and we put them into a PCA, it would make sense for FA and MD to separate into different components because they're sensitive to different features. So there's a lot of overlap between FA and MD in terms of the microstructural sensitivities, but they are slightly different. And I think seeing that play out in a component analysis, like the example I gave today, I think that makes sense as well. So I would say both can be true at once.
Is there a statistical way to identify areas of convergence between diffusion metrics along the tract? How would you do this analytically?
Good question. I would refer you back to the paper that I had on screen by Chamberlain et al. I think it was 2018, where they. Sorry, can you repeat the question?
Sorry, I just.
I got lost in my own thoughts.
I just checked it off, but it was about convergence of different metrics along the tract. Is there a statistical way to identify that?
Yeah, so that's exactly what they did. They took their major white matter tracts across the whole brain. They did a long tract analysis and put all of those, FA and MD and other diffusion-based variables in profile along the whole tract into their principal component analysis to kind of quantify, rather than just mean FA or MD or what have you in each tract. They use the along tract analysis and toss that into their component analysis to kind of increase their regional specificity and also increase the number of data points they had for their principal component analysis. So yes, it is possible to do this along tract, and I would advise that is a really great place to start, absolutely.
Are there challenges when using non-diffusion metrics in or along diffusion-based reconstructed tracts?
Good question. Yeah, I think it's yeah, that's an interesting one to kind of turn around. But yeah, I think that's reasonable, a reasonable point of concern, or at least something to take into account when you're commenting on non-diffusion-based metrics in our tracts that we've reconstructed using diffusion-based scans. But I think that's okay because it's using non-diffusion-based metrics is kind of a way for us to extend beyond the diffusion sequences, and ultimately in that example you could just think about how we're just using diffusion-based sequences to come up with a mask or a region of the brain that we're confident that tract exists in, and you can kind of consider the quality of your diffusion data and how that affects your confidence in the mask of the tract that you're interested in. But once you have that tract mask and you've defined your region of interest, I think it's reasonable to look at any imaging metric that you would like to in those tracts as kind of a way to just a way to identify the features of that tract, or at least of those voxels, which we presume are including some streamlines from that tract. So certainly not the case that we would maybe get the same results if we found a way to identify tracts using a different modality than diffusion. But I think because diffusion is such a cornerstone of this field, and it's also the body of the literature that's best represented, using diffusion to and moving beyond that allows us to connect to previous literature and really aid our interpretation, because a lot of these metrics that I had examples of on the slides, aren't used nearly as much, and so interpretation becomes more difficult. But if we're able to kind of connect to previous trends, we can really inform, our commentary in paper discussions and things like that.
Okay, 2 more here. There are many dimensionality reduction approaches. Which would you recommend?
I have more experience in applications than I do in statistical methods. So perhaps there are others on the panel that can give more informed opinions than I. My experience with principal component analysis was quite positive. It was relatively easy to implement, and I tend to prefer taking steps beyond DTI with fairly intuitive and understandable methods, because it's easiest to interpret and kind of slowly build onto our DTI-based foundational literature. Another one that I think has some promise is latent factor analysis, which kind of gets at the same idea of looking at shared features across metrics and providing us with latent variables that represent those features. I would like to hear more from others about methods like machine learning or more data-driven approaches. But I think a limitation of those methods, at least in the context of where we are in the field right now, is that we are still working on constructing large enough data sets across the developmental age range to really take advantage of those methods. So I would say the only way to find out the strengths and weaknesses of each of the, I agree, many different kind of dimensionality reduction strategies is to try them. And so far the most common method that I've seen in the field is principal component analysis, but that means there's room to explore others, and perhaps papers using other techniques would be really informative to see whether those techniques agree or diverge from our PCA-based approaches.
Okay, last but not least, could there be observable trends in shape metrics of tracts that could complement diffusion metrics?
Very interesting. Yeah, so I'm learning, I'm still learning about shape-based metrics, including from talks today. So I think that's a really interesting question. I would say, based on the other modalities of metrics that are not diffusion-based that I've seen results in or seen commentary on, I think it's reasonable to expect that shape-based metrics could also be quite informative in a different way than, say, something like magnetization transfer or water fraction or something like that. Again, kind of coming back to my answer for the previous question, because these are maybe less used in the field, the best way to move or to answer that question would be to include them in analyses with FA and MD and see if they provide convergent or divergent results. But unfortunately, I'm not too familiar with that specific literature, so I think I'll leave my answer there. But I think that's another, the more metrics we have to quantify white matter microstructure and tract structure, both big and small scale, the better to clarify what we already know about white matter development.
Okay, thanks very much.
Thanks.
Our last talk is also recorded, so we won't unfortunately be able to have live Q&A, but again, I'd encourage you to contact the presenter for follow-up questions if you have any, and I'm sure the contact information will be in the talk.